Hi, and welcome to lesson 0.4. This is on the scientific method and experimental design. These notes are a little different from what we've been taking this week. These are called doodle notes. You'll actually have 4 pages of these. These notes are probably going to be a little bit longer, and for that, I'm sorry, but this is a lot of information, okay? I have highlighted in pink what you need to fill out in your notes, hopefully help you out a little bit. And these are Cornell Doodle Notes, which means you'll be writing a question on the side here, okay? And then, like, all the notes you're taking are kind of answering that, all right? Okay. Topic question number 1. Again, we're writing that down here on this side, okay? And we are writing down this right here. What is a testable question? All right. So science is driven by questions about natural phenomenon and the way things work and behave. Questions come in 2 basic forms. They're either going to be testable or non-testable. Okay. We're going to talk about testable first. So these testable questions have a specific manipulated and responding variable, or in other words, one variable that is changed and one variable that is measured. So I've got some examples here. I only want you to write down 2 of them. Pick the 2 shortest ones, okay? So an example of a testable question would be like, how does blank affect blank? For example, like, how does the amount of water affect plant growth? Or we have, will changing the amount of soil in a pot cause the plant to die, right? We're looking at cause and effect situations. Doing something causes something to happen. All right, then we have untestable questions. So these are questions that do not indicate specific variables to test or measure. So an example would be like, why do blackberry bushes have thorns? Okay, there's no way for us to really test that question. All right, now we can turn that into a testable question. We can say, how do the number of thorns affect how many animals eat from a blackberry bush? Okay, that part right there, guys, you're going to be writing down right here. Okay, try to squeeze it in. All right. Topic question number 2. What are the types of variables in an experiment? All right, this can be confusing, so make sure you're paying attention, and we're going to talk about it some as a class as well, okay? So we have our independent variable, okay? When you're independent, that means you don't rely on anyone else, okay? You do your own thing, okay? Another way we discuss independent is by saying that it's manipulated, meaning that you're like messing with it, you're changing it, okay? So for example, we have, how is mushroom height affected by the type of fertilizer applied over 1 week? That's our experiment. So we're going to look for our independent variable in that experiment. So it's the type of fertilizer, okay? That's what we're changing, okay? It is the cause of something, okay? It is the variable that the scientist changes. So think about it like, if you're running an experiment, what are you going to be tinkering with to see those results from it? Okay, that's going to be your independent variable. The height of the mushroom is the dependent variable. Okay, it is responding to the type of fertilizer. This is the variable that scientists measure. Okay, so independent, we manipulate. Dependent is the measure or the results. Okay, one way to help remember this is if you can remember dry mix. Sorry, I always say it dry mix. I know it's up there, mix dry. Doesn't matter what order you put it in. So if we go with mix first, M for manipulated, I for independent, X axis, because we're also going to learn how to graph these. Okay, dry, D for dependent, R for responding, Y for y-axis. All right, here's an example of how we would graph this data. Okay, so at the top we have our graph title, how mushroom height is affected by fertilizer type over 1 week. Okay, it tells us exactly what we're looking at. We have our dry over here to help us remember. Remember Y, that goes on my y-axis, so that's the up and down, and that helps me remember it's the DRY. Okay, so my dependent goes over here. So what did we say was our dependent? It's what we were measuring. So we were measuring the height of the mushrooms, okay? So I've got mushroom height along my y-axis, and then my mix along the x-axis. My x in mix is for x-axis. Okay, remember M is what we're manipulating. What are we changing to see what it does? Okay, we are changing the type of fertilizer. So I've got my 3 different types of fertilizer down here on the x-axis, and you would just graph them based on the data you collect. All right, topic question number 4. Hold on, I feel like nope, okay. Sorry, I thought we skipped one. I didn't realize that the graphing was number 3. All right, so topic question number 4. What are the constants in an experiment? Okay, so again, we have to start with a testable question, something that we can actually get data on. So if we say, which fruit out of oranges, apples, or avocados will attract the most fruit flies through a hole in a jar? Okay, we can test that. So the constants are also known as the standardizing variables, meaning these are the things that stay the same. Think of the word constant itself. If something stays constant, that means it's not changing. So it's the same throughout. So we need to make sure that we are only changing one thing at a time, okay? If we're testing, okay, which fruit out of those 3 will attract the most fruit flies through a hole in a jar, so I've got my 3 fruits, okay? Maybe I have different size jars or different size holes in the jars, or maybe the apple is rotten and, the orange is fresh. So we want to make sure as many things as we can get the same from one part to the next, we keep it the same. Because like if I have different size holes, how do I know that the flies just don't prefer to go through the bigger hole? Okay, maybe they don't like oranges as much as apples, but it was hard to get to the apple. Okay, so constants are those things we keep the same from one part to the next. All right, so here, okay, we're looking at that experiment we just talked about. So we have the number of flies added to the flask to begin with. We keep that the same. Each jar is getting 100 flies to start off with, okay? Our flask is then attached by a narrow tube and small hole into a jar. We use the same size jar, the same size flask, okay? We have the same number of flies. Same, okay? The number of flies counted on fruit at the end of time. So we've got the same amount of time, okay? That's important. Again, that's a constant. And then we can see, based on these results, that our avocado is actually the most appealing to the flies. Okay. All right, topic question number 5. What is an experimental control? This one's a little bit harder to get, I think. So a control is used to evaluate whether an experiment is working. So it is usually one test of the independent variable that is like a normal value or a reference point. So if we go back to the type of fertilizer affecting plant height or mushroom height, my control would be how high the mushrooms grow in water, right? It's almost like the absence of what we're testing. Because how do I know that mushrooms don't just normally grow this tall? Okay, so I would want to compare it to a situation where that variable is gone. So again, it would be like comparing it to how they grew in water. So then I know that the fertilizer is the thing that made them grow taller. Okay. And again, it's used for comparison because if I don't have that comparison, do I really know that my difference was due to my variable? I don't, because what if that's just the normal for that situation? So we want to find the normal and then test our variable and compare them. Okay. All right. So we have our testable question here for our example. How will changing the air pressure inside a soccer ball affect the distance that the ball will travel when kicked? Okay. In the World Cup, the ball must be between 8.5 and 15.6 psi, which stands for pounds per square inch. So the test will include a lower pressure, so we got 8, and a medium pressure, which is going to be 12, and a higher pressure, which is going to be 16. Okay? Those are the 8, 12, and 16. The PSI is going to be our independent variable. That's what we are manipulating or changing in order to see what it does. Okay. How far the ball goes is going to be our dependent variable. That's what we're measuring. Okay. All right. So we got that. All right. So the distance that the 12 PSI ball travels will be the control because after researching, we found that this is the most common pressure of a ball used in the World Cup. Okay, so this is the normal situation. This is our control. This is what we're going to use to compare our 8 psi and our 16 psi to. Okay. All right, topic question number 6. What makes an experiment fair and valid? Why do we need a fair experiment? What I mean by that is, like, can I trust the results of my experiment? Am I sure that it was carried out properly? That, things weren't, manipulated or changed or, that you forgot a major step, okay? So there are a few things we do to make sure that our experiment is fair and valid. We want to trust in our results. So we want to make sure we take precise and accurate measurements. These big pink blocks here, just summarize those as best you can. You do not have to write those word for word, okay? If you're measuring, how long something is, okay? And I get out my ruler, which word ruler? Okay, yeah, I could measure in inches. I could say that, okay, this is a pen. My pen is about 5 inches, but that's not very accurate. So what I would want to do is I would then like to go down to the smaller marks and say that my pen is 5 inches and 1/16, 5 and 1/16 inches. Okay, that is an accurate measurement. Okay. So we don't want to kind of guess and get it close. We want to be as accurate as possible. We also want to repeat our trials. How are we supposed to know that our results are actually what's supposed to happen and not just like a fluke? How do we know we didn't just get lucky and get those results? So we want to do it over and over and over again to make sure that we get the same results repeatedly. Also, sometimes we just take that data and average it together depending on what it is to get our conclusion. Okay. Finally, we want to make sure we're taking detailed notes. The whole point of going through an experiment is to be able to share your data. So you want to be able to make sure that someone else really understands what you did. So we want to make sure we have our testable question clearly written, our variables identified, our materials, procedure, and data should be recorded. We're not using a journal. We'll usually have lab sheets. Okay. Again, we want someone else to be able to repeat this experiment. All right, topic question number 7. What is the scientific method? This should be review. You've had this many times, okay? Now, the number of steps in the scientific method does vary. It depends on your source, okay? This is going to go through quite a few, but it breaks it down into all the little pieces, okay? It does say to kind of summarize each one of these. So again, summarize. I didn't highlight all of this. So you guys can do this part, okay? So we start with an observation. We're using our senses. I notice something. I smell something weird. I see something that's funny looking, and it gets me thinking. What is up with that? Essentially, and that's where we start. We get our testable question. Then we're going to do some research. Okay, what do we already know about the topic? Has someone else already done the experiment? Why waste your time if someone else has already done it? Okay, use their data. And then after we make sure we're good to go with our research, we plan our experiment, okay? We got to determine our independent variable, our dependent variable, our controls, and our constants, okay? We got to hypothesize, what do we predict our results are going to be, okay? So hypothesis is a prediction of the outcome. Then we can do our experiment, okay? Back it up real quick. Prediction, pre, comes before. So we got to make sure we do our hypothesis before we do our experiment. Okay, it's kind of in the definition. Experiment, you're going to repeat trials, take good notes. Okay, you're going to be collecting the data as you're experimenting. Again, with those precise and accurate measurements. After you collect the data, you want to analyze it. So the difference between collecting and analyzing, collecting means you're writing down the information. You're writing down the numbers. You're writing down, your observations. Analyzing means that you're actually looking at that data put together. Oftentimes we'll put it into a graph or a table so that way it's really easy to look at it and see it right away. And we're going to look for patterns, right? We're going to look to see if anything stands out. Okay, so we want to analyze our data, see what it's telling us. After we figure out what our data says, then we can draw our conclusion. Okay, we're going to look back to our testable question and our hypothesis to see if our outcome of our experiment matched. Okay, remember, it's okay if your hypothesis is wrong. It's just a prediction. And even if it's wrong, you still learned something from that experiment. That is the key there. We just want to learn stuff. Okay. After we draw our conclusion, we can report our results so that others can learn from them. A lot of times, like in higher level learning or in academics, things like that, scientists will publish their work in journals, scientific journals. Okay, so for us, it's really about writing down our results on our lab sheet. All right. Keep in mind that this process is not set in stone. So put a big arrow from draw conclusion back to plan, because when the results of an experiment are not conclusive, you may end up replanning the procedure and re-experimenting. Okay, you might see that, oh, this is interesting. This isn't what I thought would happen. Maybe this changed it, and you kind of start from the beginning again. Okay, so it is constantly moving around. All right, make sure you sum it up at the bottom of those notes. And that concludes our experimental design and scientific method. Make sure you go back to Canvas and work on assignments for 0.4. We are going to be doing labs to practice these things we learned today, okay? So I will see you guys next time, and have a good rest of your day.